Hi, everyone. Good morning, and thank you for joining us in this hybrid oral session, the geochemistry and habitability of alkaline hydrothermal vents on Earth and beyond. My name is Arlene Sanchez. I am a graduate student from Stony Brook University, and I'll be chairing this session along with my colleagues here today. We have 5 great talks to look forward to today, 2 of which are from our invited guests, Dr. Deb Kelly from University of Washington and Dr. Alexis Templeton from the University of Colorado Boulder. As a note, after each talk, we will have a 4-minute Q&A where both the in-person and online audience will be able to ask their questions, so please feel free to participate. I'll go ahead and let the rest of my colleagues introduce themselves as well.
Hi, everyone. My name is Holly Rucker. I'm from the University of Arizona, and we are hybrid today. So when you come up for your question, please state your name and affiliation, and I'll also ask the same for people on Zoom, and we'll try to alternate between in-person and Zoom questions.
Hi, everyone. Thank you for coming. I'm Roy Price from Stony Brook University. I can barely see you with all the lights. But yeah, I'm a specialist in hydrothermal vents, and in particular shallow sea hydrothermal vents. And some of those are alkaline in nature, and that sort of started the ideas for this session. I'll also introduce Dr. Lori Barge, who's also a co-convener here. She's not here at the moment, but she's going to be in and out. She's from Jet Propulsion Lab in California. And I think that's it.
Great. So we have our first speaker today as Dr. Deb Kelly from the University of Washington.
Thank you for the opportunity to present some summary of the Lost City work that our team has been doing and how it can help inform us about considerations in the exploration of life elsewhere. Lost City is one of the most amazing places on Earth. It's on 1 million-year-old mantle rocks. It hosts the largest hydrothermal vent structures ever found, reach 60 meters in height. They're all carbonate limestone. Venting is medium low temperature, 40 degrees to about 96 degrees. And one of the spectacular things about this system is it's very different than other environments because it vents high pH fluids that are very low in silica and contain no metals. The fluids are also enriched in hydrogen, abiogenic methane, and formate, and other low molecular hydrocarbons that are a hallmark of fluid hydrothermal systems driven by serpentinization reactions. The fluid chemistry is strongly linked to the microbial communities that thrive within the interior walls of the carbonate towers as well as on the outside. And the system is fueled by serpentinization reactions and dominantly by cooling of the crustal rocks that form the Atlantis Massif. Dating of the limestone chimneys indicate that this is a very long-lived hydrothermal system that's been active for at least 150,000 years. As I mentioned, the reason that Lost City is there is because of a long-lived attachment fault that has exposed ultramafic rocks bounded to the south by the Atlantis Transform Fault and by the east by the Mid-Atlantic Ridge. The Lost City field itself is near the top of the Atlantis Massif, which is a very flat-topped mountain. It has no volcanic rocks. All the volcanic rocks are to the east that are on the Ryder blocks to the east on the detachment fault. So the core of the Atlantis Massif is highly altered peridotites and gabbros. And again, one of the important things about when we think about where to look for other lost cities is it's bounded by the seismically active transform fault, which keeps fluid circulation going. And also the second point is that the heat is largely derived from residual heat within the Massif rocks itself. It's one of the most beautiful places, I think, on the planet. This is video taken during a program that we did with NOAA and Bob Ballard's group. And this shows a parasitic chimney called the IMAX flange or IMAX tower that rises 3 stories outside of the main, growing from the outside of the main Poseidon structure. My student, Chris Ludwig, did dating, uranium thorium dating of the multiple samples recovered from Lost City, and she documents that this system is very long-lived with active venting over 150,000 years. This is a underwater upside down waterfall, one of the flanges. I think it's quite spectacular. It's one of the samples, the areas we sampled for microbial communities. But you can see that these are diffuse flow fluids. They have no particulates, no metals. And when we consider where to look for other lost cities, this is going to be important in how we explore for them. I won't go over the serpentinization reactions, but only to say that it's very clear that serpentinization, exothermic serpentinization by itself is not enough to drive the heat. There's not enough heat coming from that to drive the hydrothermal systems that we see. And the second point is that associated with serpentinization is a very large volume increase, up to 40%. And so this is important when we think about how we keep lost city type systems active, because this increase in volume can both expand to close fractures, but it can also open fractures. Work by Gretchen Fruh-Green shows that the basement temperatures in this area are about a little over 150 degrees C. The hydrothermal fluids, people know now, are marked by, they're very unique. There's virtually no CO2 in these fluids, and the high hydrogen and methane is a hallmark of serpentinization influenced fluids. The other two places that I've shown here for context are Lagachev and Rainbow, which are hybrid systems that include both Gabroic influenced reactions as well, high temperature Gabroic reactions, as well as influences by serpentinization. And these fluids are 360 degrees C. They have very enriched metal concentrations, but they also have the hallmark of serpentinization, which is high methane and high hydrogen, but they also have very high CO2. The mineralogy of lost city is very, very simple. It's basically aragonite that is metastable, so it converts to calcite over time. And then as seawater mixes into the porous chimney walls, forms brucite. And this is just to say again that I'll go over the hydrocarbons in a few minutes, but they are both abiogenic and look to be biogenic sourced. This is work by Susan Lang. This is part of her PhD work. And as you can see, the formate values are quite high, 36 to 158 micromolar, and acetate is 1 to 335 micromolar. So they are elevated with respect to other environments. Compound-specific carbon-14 analyses indicate that the formate is mantle-derived, that is, it's abiotic in some areas, and it fuels sulfate reducers. The acetate, however, may be a microbial byproduct of formed during fermentation of larger organic molecules. The chimney walls are incredibly porous. They're spectacular to see when you cut them open. The outer chimney walls, where there's diffuse venting, have very thick, almost kelp-like beds of thick microbial mats that are waving in the currents. But as you go into this chimney, you can see that that it's incredibly porous. This is a thin section of near the outside of a chimney. And for scale, it's about 1 inch across, but you can see this very anastomosing complex structure of carbonate. My hypothesis is that in this case, it was filamentous bacteria when seawater, the high seawater bicarbonate mixes with the high pH fluids, the bicarbonate drops out and forms a carbonate. So my guess is that these are basically fossilized filamentous bacteria that you're seeing in this one image. It's a great environment because it's a high energy environment with a lot of hydrogen, but also mixing with seawater sulfate. And so they supported a large variety of microbial communities. The porous interior walls, however, are dominated by archaea biofilms that are dominated by methanosarcinales. This is work by Billy Brazelton. I'm really proud of this paper. It's a 2010 PNAS paper that Billy and Chris Ludwig did, and they combined the dating that Chris did with 454 analysis. And it shows, in this case, his paper also talks about bacteria, but this is just results of 454 analysis on the archaea, and it shows that they have adapted to environmental change over 1,200 years. The oldest chimneys, which is extinct, are dominated by low temperature anaerobic methane oxidizers, ANME1, while the youngest chimneys are dominated by lost city methanosarcinales. These are high temperature anaerobic methane metabolizers. One of the interesting results of Billy's work is he shows that the rear frequencies, referred to as the rear biosphere, are more abundant in the younger samples corresponding to environmental change, and they can become dominant members of the communities as the environment changes over time. I would love to go back to Lost City and core these, similar to like when you're coring for to make sugar out of maple trees, core into the structures and do sequencing from the oldest to the youngest. And my guess is Lost City's probably been active for more than 200,000 years, so it'd be a great study to pursue. So challenges in this exploration for Lost City. As I mentioned, the fluids are, in contrast to black smokers that have a lot of particulates, metal sulfides coming out they look like fire hoses and very thick, dark plumes. The challenge with Lost City is that the fluids are low temperature. They're dominated by diffuse flow. And so the rise heights of the plumes are not very large, less than 25, 50 meters, certainly a lot less than 100 meters. And they're absent in particulates. So how do we go about investigating new lost cities? And this work is shown two different ways that we've done it. One is by vertical casts from a ship using an instrument package that measures conductivity, temperature, and depth, but also allows fluid sampling at discrete depths. And this just shows the plumes from discrete sample bottles of hydrogen, and you can see that they do not rise very far above the top of the massif. In a more recent study that came out of Billy and Susan's cruise, the ROV Jason was flying about 5 meters off the bottom, this lower image here. And EH is one of the best ways that you can detect these hydrothermal plumes, the oxidation reduction potential. And this shows the temperature in blue, the water depth of the vehicle, and then EH is shown, or oxidation reduction potential is shown in red here. And what you can see is that there's a very strong anomaly right where they also imaged a carbonate diffuse flow site. So this is a great way it's probably the best way, the most easiest way to detect a hydrothermal system. But it means that in other areas, in order to discover these systems, you've got to take a autonomous underwater vehicle and fly it very close to the seafloor. So there are definite challenges in how we look for other systems like this. Finally, just a summary of some of the considerations when we look for new lost city mission considerations. One is you really need an ultramafic system with active faulting. It's got to keep the flow channels open and propagation of fractures down into the ultramafic rocks so that you keep reacting with olivine and other minerals there to get the fluid chemistry that we see, the high pHs. The heat source exothermic serpentinization reaction is not enough to drive the lost city hydrothermal systems, but you really need some residual warm crustal rocks in order to make sure that the heat source is there and it's going to be active for a long enough time. And finally, one of the biggest challenges that I just mentioned is that the low rise height of the plumes and lack of particulates really requires that you fly very close to the seafloor. And as I mentioned before, the whatever the vehicle is that you are going to use, that you have an EH sensor on it, because it definitely is the easiest and best way to pick out the plume anomalies. So thank you, and I look forward to your questions.
Okay, we can start the Q&A session.
Hi, Deborah. Sanjay Som, University of Washington alumnus. Thank you for your presentation. I've been looking to share the story of discovery of Lost City because, as you said, it's such an impressive place, but I haven't been able to find the story of the discovery from your words, from your perspective. Have you published that?
No, I think there was some video things that were published. It was in 2000. The Atlantis Massif was one of the first mapped large detachment systems that had the characteristics, the lineations that we see and we were on an Alvin dive. We used Alvin, and then at night we would do camera tows up and down the vertical cliffs to try and get a better photomosaic and get a better picture of the south face of the Massif there. And towards the very end of the cruise, of course, around midnight, Gretchen Frew Green came flying into my cabin and said, I think we found something. And the camera was an old system, just black and white, and towed around with the ship you couldn't really drive it around. And so we went running into the van because I'd done a lot of hydrothermal work. And it was very clear that we didn't have any map or anything that time. We were just using sonar. So we mapped, we spent several hours till the wee hours of the morning looking at the black and white photos, and then did the follow-on Almond dive. And then the next day, Jeff Carson and I dove. We landed in we didn't have very good navigation. Took us quite a while to come into the field, and we'd driven around a lot. So we only had I think 4-hour bottom time. And at the end of the dive, we were out of power. And I said, we didn't know it then, but we were up at the top of Poseidon, the tallest structure. And I said, well, let's just float down and see how tall this chimney is. And we just kept going and going and going. And it turned out it was 60 meters. So, and then, these chimneys were white, bright white. We were sitting there on the ship, the Atlantis, the mothership for Alvin. And then on the Atlantis fracture zone and Atlantis Massif. And so I was thinking that these things look like Roman columns or Greek columns. And so that's why we called it Lost City. So the short version. It was phenomenal. It was phenomenal. It was really cool.
It's amazing. I would love to encourage you to write that story down with words and share it. We'd love to do that.
Okay, I can do that. Thank you.
Hi, Morgan Cable, JPL. Fantastic talk. I had a question about the particulates that you mentioned. You said that they don't rise very high. It was on the order of like hundreds of meters. And the reason I'm asking is because at Enceladus, in the E-ring around Saturn, we see these silica nanograins that we believe are derived from the plume and from the ocean. And so I'm wondering if you can comment on how particulates or flocculates and things that might form in these hydrothermal systems might make it up to the top of the water column and potentially be expressed in a plume of an ocean world.
Yeah, that's a good. So, I don't see though. Well, for Lost City, there's 0 particulates, and so the only signature we have is from the chemistry, right? But in black smoker systems, certainly the rise heights are, they can be a few hundred meters off the seafloor. I would guess we don't really talk too much about currents, right? But depending on the topography and the currents, there's one way you can get them up there is if you have currents, but it all depends on the density structure, right, of whatever that overlying water body is. So.
Thank you.
Hi, Deb. This is Chris German from WHOI.
Hey, Chris.
You made a really important point, I think, about the need to come up with clever ways to actually search and find more lost cities. But the other thing I was going to point of, like, given how much stuff we continue to find, how often do you think the conditions for a lost city might recur on our planet, and how many more lost cities might be out there? It's going to be hard to go find them, but do you think there's just 1 or 2, or could they be free?
No, I actually think they're ubiquitous, right? And the reason I say that is particularly in slow and, as you know, in slow and ultra slow spreading centers, where we see these transform ridge intersections. These massifs are very, very common, right? They're a hallmark. It's just that people have always focused, on where the black smokers are, right? And so to drive 25 kilometers off axis, we tried to get, Jeff Carson and I tried to get funded 3 times to go. There's another massif absolutely identical to the one that Lost City's sitting on. But that wasn't successful at the time. So I think they're very common. I don't see any reason why they wouldn't be, right? Based on all the characteristics of the ridge transform intersections. So
I think that's a good take home, right? Is like, they might be hard to find, but there's probably plenty of them, so it's worth a try.
yeah, thank you.
Thank you. Our next speaker is Antonin Affolter from IMCCE in France.
Good morning, everybody. So my name is Antonin. I'm finishing my PhD right now. The manuscript is due tomorrow in Paris. Pardon me in advance for the messiness of this presentation. I'm a bit overwhelmed at the moment. But I will try my best to talk about this project that I worked on during my PhD work, which is about using ecological modeling to constrain the habitability of a potential hydrothermal environments in Enceladus's ocean and try to discuss how such a hypothetical biosphere that could be there could be detected. Right, so Enceladus, and thank you, Dr. Cable, to actually mentioning a little bit about Enceladus, sparing me a few seconds of introduction. But it is this moon that orbits around Saturn, most of you know, but it has a liquid water ocean that is overlain by an ice layer that has cracks, and a space plume erupts from these cracks. And this space plume was visited by the Cassini spacecraft. And the Cassini spacecraft found in this plume some very interesting gases, such as methane, hydrogen, and carbon dioxide. And these are evocative of hydrothermal systems, just like the one that was described by Dr. Keeley, which is Lost City hydrothermal vents. So alkaline vents where serpentinization fluid is released in the ocean. And yeah, as mentioned, these vents are populated by microbes, such as this one here, that rely on the conversion of hydrogen and carbon dioxide into methane as an energy source. So it is very interesting that Enceladus has, not only a liquid water ocean that, makes us think about potential habitability, but it also has a very specific environment candidate to habitability. And not only candidate environments, but also a candidate type of organism in these critters here. So what we did as, ecological modeling biologists, we said, okay, well, we'll try to model this type of organism and see if we can have a definition of viability of a population of such organism that can emerge from a model, and also try to quantify how much methane would be produced by such population, and try to put that in comparison with what was measured by the Cassini spacecraft. So we went on and designed a model of population dynamics, where we focused on the energy yield of the catabolic reaction. The catabolic reaction is this one, right, that produces energy for the cell. You see that in this model, we don't have a classical way of modeling the availability of nutrients and stuff like that. We look at the energy yield from the catabolic reaction. That's the variable that is important to us, because these organisms that rely on this type of reaction have very low energy yield reactions, and they operate close to thermodynamic equilibrium. So in modeling that, we input parameters here that kind of fit the organisms that, such as this one, Ankylosus, in order to have this pattern of survivability to temperature, for example, here, where our modeled strain does not survive above 90 degrees. This is an important point, of course, because there might be liquid water at temperatures above the temperature of 122 Celsius, at which is the maximal temperature at which we can observe growth so far for any organism. And then looking at the population dynamics, so the way the population grows over time, we look at that, and we look at the affinity of the catabolic reaction, or minus the Gibbs free energy that is here. And that depends on the abundance, the relative abundance of the hydrogen, methane, and carbon dioxide. And we see that as the population grows, this energy yield affinity goes down until a certain limit. At this limit, the population is at steady state. It doesn't grow anymore, it doesn't die off. And this steady state is characterized by this term in the parentheses here being 0. So the Gibbs free energy is such that everything here is 0. This is an interesting point to make, because if the initial state of the system was below this affinity, then growth wouldn't have occurred, because this term here would have been negative. In a way, that measures the viability of a population of methanogens, such as those that we have modeled in a given environment. So what is left to do now is to model said environment. So here we now go into the physical modeling world, and we try to get a completely idealized vision of what is the environment around a hydrothermal vent. So we have hydrothermal fluid here that is coming from, well, the ground and is mixed with ocean water in the so-called mixing layer. And then this mixing layer has, some buoyancy because it's warmer than the rest of the ocean, and it starts rising and forming a plume. And this plume actually is thought to reach the icy layer in Enceladus because of the way the ocean of Enceladus is probably not stratified as the oceans that we have on Earth are. So then we model our population, or the habitability and the potential population in this idealized environment. And here on this figure, we can see around the hydrothermal vent how the concentrations are modified by the action of a population. The blue line represents the hydrogen, which is completely depleted here in this particular simulation. Methane is produced in green, and so on. And we can connect that to the composition of the plume that rises above the mixing layer. An important remark here is that we consider the population to be fixed in this mixing layer. It does not travel with water that rises across the ocean. So as Dr. Kelly mentioned, the microorganisms that are found in lost city hydrothermal fields are found in biofilms mostly, so they are fixed, they are localized in the environment. Okay, this is just to keep in mind for later, but in the first project, we looked at how these modified concentrations could be translated into composition of the space plume, and how we could compare different scenarios regarding the habitability of the hydrothermal vents to the measures carried out by the Cassini mission. And this work was published last year, so you can look at it. So we simulate these composition and these hydrothermal vents for quite a wide range of parameters and conditions that are attempting to reproduce, uncertainty or variability in the composition of hydrothermal fluids here on Earth. So, we take the hydrogen concentration in hydrothermal fluids from, very low to the maximum in suboptimization experiments, and we do the same for methane, for example, and temperature and stuff like that. So here we have distributions of simulations under three scenarios. So the first one is the blue line here, which represents the uninhabitable simulations, where nowhere around the vent the conditions were right for the population to grow. So these are the blue ones. The orange ones are where the conditions somewhere around the vent were found to be habitable or viable to a population of methanogens, but we just left it abiotic. And then the green one is with biotic production of methane and consumption of hydrogen. Looking at the distribution for hydrogen escape rate in the plume compared to the magenta lines here that are the observations made by Cassini, we see that the observations, they match with the hypothesis that there might be, a hydrothermal environment that is viable to the modeled population of methanogens. And not only that, but also the depletion of H2 by the methanogens is not sufficient in our model to really make a difference, and it cannot enable us to conclude that methanogens are unlikely based on the abundance of H2. So the observed abundance of H2 to us for our modeling is compatible with both habitable conditions, but also conditions where H2 has been depleted by methanogens. And then looking at methane, the abiotic distributions in blue and orange, they do not match with the observations. So our modeling suggests that serpentinization, which is the only abiotic process that we consider here, is not a good explanation for the methane that was found in the plume. But under the hypothesis of a methanogenic population, the observation is kind of likely, right? So based on these quantitative assessments of the production of methane and consumption of H2, we cannot really discard, based on the Cassini data and our understanding of serpentinization, discard the hypothesis that a methanogenic population is present. Okay, but next we looked at how this population would, look like in terms of size and stuff like that. So we tried to constrain the total biomass that would be represented in such a population. And here we have two distributions. So this one doesn't assume anything regarding the methane output of the population, and the dotted line is the distribution under the assumption that methane in the plume comes effectively from a population of methanogens. So this is a subset of populations that produce enough methane to explain the observation best. So we find this population to be quite small, about the same mass in terms of carbon equivalent to one single whale, orders of magnitude below the biosphere archaea on Earth, or even humans. Also, the important point about the biomass being fixed. So here we are considering the total biomass, which is localized in an environment. So, for example, this is a representation of the density of biomass around the fluid. But we are around the hydrothermal vent. But we are interested in what we can potentially find in the space plume, and even before that, the plume that rises from the hydrothermal vents. So for that, we have to consider the biomass production, because this is the quantity that scales with whatever can escape the system. So we computed that as well, and see that the production normalized by the production of hydrothermal fluid that we expect for a population that produces the methane that is found in the plume is quite close to that of hydrothermal ecosystems on Earth. And finally, my time is running out, so I'm going to be fast. But finally, we would like to constrain, how could we design the mission, or help design the mission, so that the risk of false negative is constrained. What I mean by that is that we'd like an instrument to have sufficient resolution, or we'd like to capture a sufficient sample, so that the risk of not seeing a biosignature in the case where a population exists is minimized. So this is the meaning of this figure here, where we're trying to assessing the minimal sample volume size that is required, so that if there are dead cells in the plume, they are captured. And here on the right side, we're looking at a molecular signature, such as glycine, that can be produced abiotically and biotically. And we kind of map the concentrations that would be evidence in favor of abiotic production or in favor of biotic production. So yeah, I'm gonna leave it here, and thank you very much for listening. Thank you.
We have time for one question.
Hello, Morgan Cable, JPL. Great presentation. So you mentioned that the organisms that you modeled, that they just hung out down at the base of the hydrothermal vents, and they were wrapped within biofilms. So if you were to search for biosignatures in the plume, it sounds like you would focus more on the molecular ones, that may be looking for, cells themselves might not be the right strategy. Or can you comment on that a little bit?
Thank you for your question. So yeah, I mean, we enter here some speculation, because it isn't clear whether the proportion of dead cells that can leave the system as cells retaining their structure, or as just broken down cells and molecules. My guess would be that most of these cells that escape are dead or dying, because they are leaving the environment where the conditions are right for them.
Yeah, that's okay. If they, get shot off into space, they're probably not long for this world anyway.
Yeah. So yeah, the question boils down to kind of guessing whether what would be the proportion of cells that would keep some structure until the very end, or that would be under biomolecules. To me, it sounds more cautious to look at molecular biosignatures, to consider molecular biosignatures. Because even though there have been some papers suggesting that cells can retain some structure when they're outgassed in space, most of them actually don't. And in any way, we would only look at broken down cells and biomolecules.
Thank you, and good luck with your submission of your thesis tomorrow.
Thank you.
We don't really have time for another question, but sorry, my mic's not working. I encourage you to reach out to Ismet offline after the presentation.
Thank you for your questions. Our next speaker is Rabia Papal from Aix-Marseille University.
Yes, so good morning, everyone. I'm a first-year PhD student at the Mediterranean Institute of Oceanography, and today I'm going to talk about cultivating microbial consortia from the Prony Bay, New Caledonia. So our work is based on the submarine alkaline vent theory of the origin of life, which focuses on the way a cell obtains its energy. So in these hydrothermal, alkaline hydrothermal vents environments, we have very steep redox and pH gradients that create proton gradients across micropores in the chimneys. And these proton gradients are reminiscent of the proton motive force that virtually all known cells use to generate their energy. So we have a link between the inorganic and the organic here. And our team is trying to understand the microbial communities in present-day alkaline hydrothermal systems to hopefully understand, to hopefully open a window to the very early microbial communities on Earth and potentially elsewhere. So the driving force at these alkaline hydrothermal systems is serpentinization, which is the hydration of ultramafic rock, in which creates serpentine, brucite, and magnetite, and most importantly, a very large amount of hydrogen gas. It also creates a very alkaline milieu and heat. And the hydrogen gas can further react with carbon dioxide from the system in an abiotic reaction to methane and small organic molecules. But most of the CO2 that is present in the system is immediately precipitated in carbonates because of the very alkaline pH. So we can see that serpentinization creates very harsh environments for life. However, it does provide compounds for microbial metabolism, such as the hydrogen, which can be used as a large energy source, also the methane. A carbon source with formate acetate, other small organic molecules, and methane. And the ambient seawater also supplies a range of potential electron acceptors. Most importantly, in modern systems, these are oxygen, nitrate, sulfate, and dissolved carbon. So our team is studying the Prony Bay hydrothermal field, which is located at the southern coast of New Caledonia. And because the system is shallow, it comprises characteristics of both terrestrial and marine serpentinizing systems. So we might be eventually able to use it as a model system for other systems that are less accessible. And as you can see here, it comprises terrestrial, intertidal, and marine sites. And what makes it special is that the hydrothermal fluid is of meteoric origin. So we have a steep salinity gradient between the hydrothermal fluid and the ambient seawater. So our team is now starting to study the metabolic functioning of the microbial communities at Prony Bay. But the issue with that is that the majority of the key players have not been cultivated yet. So we are lacking the experimental evidence. So our aim in this study was to cultivate microbial consortia from Prony Bay for the first time. And we chose to assess the effect of the electron acceptor on community structure. So we were working on samples from 2018 that were taken from the intertidal site at Prony Bay, and we used them to inoculate 4 different culture conditions in the controlled bioreactors. And all of these culture conditions were supplied with hydrogen as an energy source, with both formate and acetate as a carbon source, and with 4 different electron acceptors being oxygen, nitrate, sulfate, and dissolved carbon to promote methanogenesis. And our experiments were all at a pH of 10 and at a temperature of 35 degrees Celsius. And we maintained them over a period of 2 to 4 weeks. And after this period, we conducted analysis. So we conducted HPLC to follow the consumption of both formate and acetate over time IC to follow the consumption of nitrate and sulfate. And we performed metavar coding to assess the changes in community structure over time. And we are also working on the metagenome and metatranscriptome from the cultures. So in all of our culture conditions, we saw considerable growth. And what was interesting was that the growth really reflected the potential energy for hydrogen oxidation with the respective electron acceptors. So we had most growth in the culture grown on aerobic respiration, followed by nitrate reduction, sulfate reduction, and methanogenesis. For the consumption of the carbon source, we could see that both acetate and formate were consumed. However, we could not really establish a clear preference for either of the compounds. So yeah, this question remains open for the future. For the electron acceptors, so in conditions, you can see here sulfate on the secondary axis, and it was not really consumed at all. However, something else was consumed, and that was thiosulfate, which was a compound of the base media. And we can also see a production of sulfate. So this consortium seemed to prefer thiosulfate over sulfite. And here in the nitrate reducing conditions, we can see that nitrate was equimolarly reduced to nitrite. So we have an incomplete denitrification. So moving on to the bacterial diversity, to the left, you can see the diversity of the inoculum, so of the natural community. And to the right, you can see the diversity in 3 of the 4 culture conditions. And you can see that only a very particular part of the community was enriched. And if we look at the Shannon index, we can see that most of the natural diversity was retained in the sulfate reducing condition, or in our case, the thiosulfate reducing condition. And yeah, this might point to a significance of thiosulfate as an electron acceptor in the natural system. Other than that, we mainly enriched Serpentinomonas, which is really a marker species of serpentinizing environments and one of the most alkalifile organisms that we know of. And we also enriched a large proportion of Anaerobacillus, especially in the thiosulfate reducing condition. Whereas it's important to know that the species of Anaerobacilli here was different from the species of Anaerobacilli here. So here you can see again that there is a huge taxonomic overlap between the oxygen reducing condition and the nitrate reducing condition, whereas the thiosulfate reducing condition is really different, probably due to the lack of Serpentinomonas. We also had a look at the archaeal diversity, but the archaeal abundance was way lower than the bacterial one, which is quite typical for serpentinizing systems. And you can see that we almost exclusively enriched methanogens. And what is quite exciting is that we were able to observe 2 species of Methanosarcinalis that are really endemic to serpentinizing systems, which were the Lost City Methanosarcinalis and the Cedars Methanosarcinalis. And we have been trying to cultivate them for more than 20 years. So it would be really cool if we were able to replicate these results. So the consortia that we cultivated might actually play a key role in the natural system. So here you can see a theory that was postulated by Lang and Brazelton on how the microbial communities in serpentinizing systems are able to overcome the extreme limitation of dissolved inorganic carbon in the system. And according to this theory, our consortia are part of a consortium of foundation species that are able to grow on small organic molecules such as carbon, formates, and acetates, and transform it into carbon dioxide, which can then be used by a larger and more diverse community of autotrophs, which in turn support a different community of heterotrophs, which end up remineralizing the CO2. And Lang and Brazelton, they really underlined the significance of sulfate as an electron acceptor within the foundation species. However, since our consortia seem to prefer thiosulfate, it would be really interesting to assess thiosulfate reduction and disproportionation in the system. especially because it is very likely that thiosulfate was abundant at alkaline hydrothermal vents in the Hadean, whereas sulfate was most likely not. So yeah, that would be a very interesting perspective. So to conclude, we were able to successfully enrich Prony Bay hydrothermal field microbial communities on 4 different electron acceptors. We were able to measure their metabolic activity, monitoring thiosulfate and nitrate reduction. However, something remained unclear, and that was the carbon source. We were enriching a very particular part of the natural community, especially Serpentinomonas and Anaerobacilli, and potentially some previously uncultivated strains of Methanosarcinalis. And as thiosulfate reduction might actually play a key role in the foundation species consortia. So our next step will be to assess the question that remains open in these experiments, which is the question of the carbon source. So we have previewed a new series of cultivation experiments in which we will follow the consumption of the carbon source using stable isotope probing coupled with nanosims and metagenomics. So thank you very much for giving me the opportunity to present today. I would like to thank Paul Lestoop, who produced these results in the frame of an internship, my PhD advisors, Gaëlle Rousseau and Dr. Anne Postek, and the rest of the team. And yeah, thank you for your attention.
We have time for questions.
Hi, Julie Huber from Woods Hole. That was a great talk. It's so exciting. I'm really jealous about your bioreactors. They're beautiful. I was curious, it looks like in the bacteria, you're really enriching for groups that have only been described from terrestrial serpentinizing environments. Is that correct? Serpentomonas has only been from land-based environments versus your archaea, which looks similar to some marine groups. Is that right?
I'm pretty sure that some of our groups that we enriched can also be found in marine environments. So it really shows that Prony Bay has the characteristics of both terrestrial and marine sites. So yeah, it is really sort of a hybrid system, which is pretty cool.
Okay, thanks.
Hi, Fabia. Sanjay Som, BMSIS. I loved your growth experiment. Super cool talk. I was wondering if you, and I may have missed that plot, the consumption rate or production rate of methane versus concentration curve to get at some of the enzymatic parameters of methanogenesis.
Sorry, could you repeat the question?
I was wondering if you, in your growth experiments, plotted the methane production rate versus concentration, or to get at the enzymatic parameters.
No, we did not do that, but we would love to follow the production of methane. So in this series of experiments, we were not able to do that, but we are really hoping that, yeah, in the next series of experiments, we can.
Thank you.
We have time for one more question. Okay, thank you, Rabia. Our next speaker is Dr. Alexis Templeton from the University of Colorado Boulder.
First in-person speaker, so. All right, thank you. I'm going to be talking today a little bit of an integration or synthesis of work we've been doing in the Oman ophiolite as a terrestrial analog for both ocean worlds, but as I'm going to talk about in ways that I think are connected to Mars and some of the recent discoveries as well. And in this work, it's going to be some aspects of science that was done through the Rock Powered Life NASA Astrobiology Institute and in close collaboration with the Oman Drilling Project. So I'll start with these cartoons. In many ways, when asked to give a talk in this session, my focus starts over here in our different perspectives of hydrothermal processes on the rocky crust of ocean worlds. And I want to talk about the fact that today I'm going to often speak to non-hydrothermal conditions, so not necessarily where a heat source is, but just the pervasive chemical reactions and habitability that's generated from ultramafic rocks reacting with cool fluids in these systems away from those heat sources. And that's why I also think it's relevant in Mars. Often when you see a cartoon of the subsurface of Mars where we think there may be groundwater storage and in contact with olivine-rich rocks, there's a sense that water-rock reactions can produce things like molecular hydrogen as electron donor. But we know that there's a lot of olivine-rich lithologies very close to the surface of Mars, and the potential for their interaction with water and to create conditions that are conducive and habitable for alkaline vent-type chemistries, I think, is very prevalent there as well. So this is the Oman ophiolite system. It's a very large exposure of ultramafic rocks rich in minerals such as olivine and pyroxene. And in these types of systems, we're really looking at fluids that are near surface temperatures. There's no heat source or tectonic activity currently, but there's extensive hydration of these rocks, and it does lead to the pervasive generation of both alkaline and hyperalkaline fluids. And in the places where we can actually see the water expressed, which often is in a seep-type environment in this kind of context, those fluids that range from pHs of 9 to 11 are very high in hydrogen, methane, and ammonia. So again, the sense that you can see that state generated at lower temperatures. And for example, if you want to look at the modeling of the kind of chemistry of these fluids or some of the biology of who lives in these seeps, Everett Schock's group has done a really beautiful body of work on this. What I'm going to talk about today is when we look at the surface environment that's particularly dry, other than these few indicators of where fluids are escaping, the deeper system is incredibly wet. It's storing massive volumes of water, and it's not only habitable, but highly inhabited. And the way we've been looking at this is through the partnership with the Oman Drilling Project. We've been able to establish a series of boreholes that range usually from 300 to 400 meters in depth that poke in and penetrate across all sorts of different windows into the subsurface ultramafic system. And sometimes those holes were drilled just to give access to the fluids that are circulating there, and at times we were able to do core recovery with excellent core recovery, with bringing back more than 1 kilometer of core from subsurface environments. And the beauty is that we can pair often the chemistry and the mineralogy of those cores against what we see in the fluid system and the alkaline fluid chemistries. So I had to show something. We don't have an alkaline vent, so this is me throwing a GoPro down a well. I literally have it attached to a fishing line. I had to duct tape a flashlight onto it. But what I hope you can see here is we're in water. We hit water within meters. And we're going down, and you're seeing the flocks of material, of particulates, of microbial biomass, and mineral particles that are present in these fluids that are circulating. And we have a highly fractured rock system here with transmissivity of fluids. Wow. My screen isn't doing that. It's stable, but it looks like you don't have one, and so I hope they'll settle it down, just because I'd really like to walk through what these chemical profiles look like when we're able to go down into the subsurface serpentinite. So I'll just pause here for a minute. It did come back. I still own the GoPro. I've thrown... And of course, there's such things as I'm waiting for them to fix it as good borehole televiewers and things like that. But when we first started working there, I just didn't have that equipment, and we wanted to see what we were starting to send pumps down into or other instrumentation to collect data. You want to reload the presentation? Escape and come back. Gotcha. Yeah. Okay. Yeah. Okay. Hopefully going past the GoPro movie doesn't instigate that again. All right. Is there anything on the screens? Yeah, I can't hear you. I'm sorry.
This is what's shown on the screen.
Okay. Yeah, again, we can see just fine up here. I could go give Dan Tolman's talk, which is going to be very good, by the way, with about the acetogens in the system. Let's try, no. Great, okay. Wow.
Maybe minimize the presentation view so it's just the slides.
Yeah, I don't have that kind of control here. Okay, so if you can find a way to do that.
Copy that to the desktop. She can run it straight through PowerPoint bypassing the system.
Ah, okay. We'll do that. I'll see about 30 seconds.
After a year of Zoom teaching, it's we will come back.
That's it, right?
No, that's the next talk. It's this one right. It's this one right here. Okay. Yeah.
Perfect.
And
All
then
right.
how are you going to now go to the next this one? How do we get it projected onto the screen? So-
As soon as I unlock the button back
Okay,
there. Yeah.
great, thank you.
So just give me a second.
Yeah. I think the moral of the story is that they just want Dan Coleman's talk to come up. So please stay tuned for that in just a minute, and I think we'll get back to data.
Show an overlay.
Maybe just talk through it. And
Yeah, I'm trying to get it to. Yeah, I'm trying to get it to come back there. It doesn't. Okay. Wow. All right. Well, in order to move forward, please ignore the data up here. I just want to talk for a moment about what was on that previous slide. Apparently, I'm not supposed to reveal the strong chemical disequilibrium in the subsurface, and it's spectacular. It's only partially shown here. So when we do these downhole profiles in these boreholes, the thing that we observe is essentially that we can move over almost a volt in electrochemical potential, but over, we might go hundreds of meters, but those transitions happen over the span of just a few meters. So we're often going from 200 or 300 millivolts to minus 700 or close there. We're sitting right at the lower stability limit of water. Similarly, those same chemical gradients are established. We're looking at pH variations that are usually going from about 8 up to above 11. And the transition is a little bit broader there in terms of maybe tens of meters over which we see those pH variations happen. And when you look at the deeper components in those systems down at 200, 300, and 400 meters, this is where we have highly reducing conditions maintained and millimolar concentrations of hydrogen and methane. There's very low water-rock ratios that are present here, and we're storing the fluids under these highly reducing conditions. And then they're in contact with a more active hydrologic regime, which is where we have nitrate, sulfate-bearing fluids that are circulating through, and some dissolve CO2. These are anoxic. So oxygen's scrubbed out incredibly quickly in these systems. The temperatures, they're only 30 to 40 degrees C. And the other component of the rogue slide is just that, remember, again, we can look very closely at the mineralogy down hole and from the cores that we've recovered. And so we're able to pair those states of the system against where we see an enormous amount of relict olivine that's present in the rocks and initial serpentinization to areas where it's almost fully hydrated and serpentinized, but we have a lot of ferrous-ferric phases, the green rusts, relic spinels, and magnetite formation. And only in the upper 5 or 10 meters do we really see oxidized conditions in the rock. So then this would be a different area where we again see pretty strong chemical gradients on the slide that I have moved on to now, where I'm saying there's a lot of reducing power left. The idea here is simply that we can go to some of the holes at the most extreme conditions of high pH, sitting at 11.5 or so, and we again land on the lower stability limit of water. There's not much relict olivine here, but there's a lot of reducing power, both from things like awaroite that were generated under high hydrogen fugacity during the water-rock reactions at these low temperatures, and the prevalence of these minerals such as brucite. It's a magnesium iron hydroxide phase. It's substituting an enormous amount of iron into its crystal structure at these low temperatures, often up to 35% of the cation component that's in here. And that iron, too, in brucite is playing a big role in the buffering the aqueous geochemistry. So to come back to the sense of not only is this a habitable environment with a chemical disequilibrium, but it is inhabited. And it's actually a thriving biosphere from every measure that we've been able to apply to this so far. So as we're starting to look into the rock cores to do life detection, for example, we're thrilled at the opportunities when, for example, we can pop out these large cell clusters that seem to be distributed into some of the parts of the fracture system, although we're very interested to know exactly where those are localized and where we'll have the best probability of finding them. In general, we've been able to interrogate both the fluid and the rock system to try and determine cell abundances. And it remains quite high through most of the system. So if you look at it from a fluid perspective, from some of the cell counting work that Libby Phones did, we're almost always at seawater type cell abundances of 10 to the 5 up to 10 to the 6 cells per mil. When we move into the rock cores, we're often homogenizing or taking a sample from a large volume of rock in which the cell densities can range from 10 to the 2 to 10 to the 7 cells per gram. But again, these are robust numbers in terms of the biomass that's being harbored within the rock matrix. So our goal right now is how to improve our strategies to detect evidence of both preserved life and current life activity in these rocks. We want to be able to answer these questions. If you pick up a serpentinite that's undergone extensive hydration, even at low temperatures, how do you prove that it was alive or is right now? In the shallowest samples that have been recovered from the Oman drilling project, where there's extensive carbonation, there's been work that's ongoing by John Zalumis and his collaborators to try and look in the carbonate phases at the preservation of filaments, iron oxide structures, and organic carbon that are preserved in there, and to interrogate them for their potential as biosignatures. In my lab, one of the things that we're focusing on more is as we move into this anoxic anaerobic subsurface biosphere system, the massive volume that really dominates this environment, how do we start to detect where there are active cells? One of the approaches that we commonly use is to use hyperspectral Raman imaging. We're often doing it at the microscale. And from it, we can pull out exquisite textures of the complex alteration history of these rocks, the different generations of serpentine that are there. And then nested in that framework, to start to look for then things like lipids that are preserved from cells or active cells that are there. So I just point you now to look at some talks there this afternoon from both Srishti Kashyap and Tristan Caro, who are going to talk in the life detection and deep biospheres about some of the strategies they're applying to do this. What we have more data about is at the bulk scale. And by bulk scale, I mean taking a chunk of rock core and being able to then put it into a series of assays, or to take biomass pumped from fluids and use them in a series of assays to measure microbial activity. And one thing that we feel very confident in now is that biological methanogenesis is prevalent. And that might not seem like a surprise, but when you do look at the geochemical state of these fluids at pH 11 and the low carbon availability, there remain many conundrums about the physiological adaptations to allow methanogenesis to proceed. Bulk chemical isotopic measures like delta 13C of methane also indicates that maybe it's potentially abiotic source. However, a series of different works that have come out in the last 2 years have come back again and again and say there's evidence for biological activity, the producing methane under these conditions. Some of them are from Dan Notaft. He's published a series of papers with isotopolog geochemistry of methane that shows the kinetic disequilibrium preserved in the methane that's abundant in these systems. Other work comes from Emily Krauss. She was looking quite a bit at both just the abundance of methanogens across different geochemical parts of the system. They often can be up to 20–25% of the total population, mostly methanobacterium species. But at times when you quantify the transcripts from those same fluids, it can be more than 65% of those total abundance. And then Libby Phones has done quite a beautiful body of work in her thesis that first was looking at quantifying the rates of conversion of 14 labeled bicarbonate into methane from different geochemical states of the system, and then trying to look at some of the diversification of the methanogens in these systems and their adaptations to growing at hyperalkaline pH. And in particular, the role of formate, as was just mentioned in the previous talk and by Billy and Susan Lang and others, comes back again and again, trying to understand the role formate's playing as an intermediate in these systems. So in Libby's work, she first started working in the fluids, looking at rates of formate conversion to methane and the pathways at which it was being used. She also moved into starting to look at the rock cores themselves. And one of the surprises for us was that when we could get a measure of formate, both conversion to methane or formate oxidation in the cores, it's sometimes several orders of magnitude higher than what we saw in the fluids. And I think we've had a bias so far that the fluid system was where a lot of the dynamics and activity were occurring biologically. But again, it brings us back to wanting to understand where and how in the rock cores themselves these microbial communities are being so active. So pivoting a little bit, we have also been focusing on processes such as sulfate reduction. This is a terrestrial system. However, sulfate's abundant through prior water-rock reactions on the seafloor and other sources of sulfate that have been stored within these rocks. So we can often have up to millimolar concentration of sulfate. And Clemens Glombica, working in Tori Holler's lab, applied techniques often used in deep sea sediments to use 35 labeled sulfate and try to measure rates of biological sulfate reduction in the fluid system and then later within the rock cores themselves. The rates are exceedingly slow. They're hard to measure. They're just above the limits of detection and some of the slowest rates that have been measured on Earth. But they are pervasive again and have been able to be measured in both systems. And so we've become increasingly interested in the importance of this slow sulfate reduction in the overall geochemical dynamics within the ophiolite. Specifically, for example, during drilling, we started to notice that often we would bring up core intervals that were degassing. You can see sort of a bubbling wet core here on the left. And when we look at these optically through a thin section, they're optically darkened. They're becoming more opaque. And the total sulfur content in these serpentinites is not high. It's a maximum of up to 0.6 weight%. But our interest is in when and how are the reactions with sulfur occurring. And to have hydrogen sulfide produced in these fluids, which also can be detected, is intriguing because at these temperatures, we're not expecting thermochemical sulfate reduction to occur. So again, we're just inferring it to be a biological process, as also shown through rate assays. We're working with Bethany Ellman and Rebecca Greenberger in another related project, where Rebecca was able to scan the entire 1 kilometer of core obtained from the subsurface to look at its spectral characteristics, and we can from that define different styles of serpentinization in different geochemical regimes in the rock core. And often she's looking at the iron 2 or iron 2-3 transitions in the serpentine in terms of the ways that she's imaging it. But the second you get to these partly sulfurized cores, the optical darkening and the decrease in the reflectance really changes the signal and what you would see. So we're zoning in on those areas within our cores and starting to look at them optically, and again, back with things like Raman or an X-ray spectroscopy, trying to characterize the sulfurization process itself. And in this particular case, with this kind of mapping, we would be able to start to see sulfide replacement of olivine mesh cores. The brucite that replaced the olivine is now being itself replaced by sulfide. And we can see reaction rims that are forming within them. And this is giving us the mineralogical context to, again, want to come back and ask the question, where are microbial cells distributed within this, and where are they active, and what's the potential imprint of their activity? if we step back out of the microscale back to just the bulk rock scale, an example of this too would be work by Katie Remfort in her PhD thesis. She came and took several intervals of subsurface cores trying to see if she could successfully extract intact polar lipids from the active microbial community. She's also done that from pumping the fluids that circulate within the system and collecting biomass there too. She has a beautifully complex story. I'm just going to take one component of that to say one of the most striking findings from our work is that despite us knowing that we have quite a complex microbial community structure, what we see again and again in the intact polar lipids that are able to be extracted and characterized from the system is this dominance of these glycodiether lipids. And part of those are archaeol that we're attributing back to Methanobacterium that's so dominant in our system, as well as these 9-isoprenoidal diethylglycerol lipids that we attribute to sulfate-reducing bacteria, which there are many present, but Thermodissulfo vibrio is one of them that we have in culture and are working with to characterize the correspondence in those lipids. And they often together, just these two, make up more than 90-95% of the total IPLs that can be obtained. So thanks for your patience through that little intermission. And in general, the part I didn't get to show you, but I'm still really excited to leave a visual imprint of is the really strong chemical gradients existing in the shallow subsurface, even of these low temperature cool systems. And strong reductants persist, which allow these to be maintained over long time scales to have this disequilibrium present and harnessed. We're seeing a very abundant biosphere, in my opinion, at these kind of high cell densities in both fluids and in rocks, greater than 10 to the 5. And when we take whole rock cores or take fluid biomass, we can definitely measure activity, turning over things like 14C bicarbonate, formate, sulfate. What we're really interested in are what are the real adaptations that are allowing the microbial life to be active in the system. And again, Dan will talk a little bit about that in terms of acetogenesis in just a moment. But in this question of how do we come recognize that the system's alive and how do we know what to target for in terms of analysis for biosignatures or showing life activity at that cellular scale, we're very intrigued by the sulfurization of these cores that's occurring, not only to be potentially a signature that this is an active system, but also because it may well be playing a role in preserving some of the lipids that are in the biomass that's present. And with that, I'll move to this afternoon's session and just encourage you to come and hear more about efforts to detect activity in specific microbe mineral associations. Thank you.
We have time for one question, and we encourage others to speak to Alexis after the session as well.
Hi, Alexis. Wonderful presentation. I was wondering if you have a sample where you're both measuring the methane production rate and the cell densities. I'm trying to tune a biomass model.
Yeah, if you look to Libby Phone's work, she did do that. So she's got the cell numbers that correlate also with the activity. Now the system stimulated because in that case, they're adding 14C bicarbonate or formate, often at a millimolar concentration, and the ambient concentrations in the system tend to be in micromolar concentration. But that's probably where you could find the best data of both the geochemical state and the cell density.
Yeah. Awesome, thanks.
Yeah.
Thank you, Alexis.
Thank you.
Our next speaker is Daniel Coleman from Montana State University.
There we go. All right. I'm not giving a chalk talk, so that's a good start. Thank you to the conveners for the opportunity to talk about some of my research. That was a great introduction to it in Alexis's talk. My name is Dan Coleman. I'm an assistant research professor at Montana State University in Eric Boyd's lab. And so what I'm going to talk to you about today is kind of extending on from the work that Alexis was just talking about in the smell ophiolite and looking at adaptations of putative acetogens that are dominant in some of these subsurface aquifer fluids. So, as many of us in the room are pretty well acquainted with this idea, and a lot of the talks in this session have really elegantly pointed this out, these serpentinite-hosted environments are really ideal analogs for trying to understand the habitats that supported some of the earliest life on Earth, as well as the potential for life on other planetary systems. One of the most important attributes of these systems are the water-rock interactions that are occurring in these serpentinite systems, and especially the production of abundant amounts of hydrogen, right? It's these water-rock interactions that produce these substrates that can support chemosynthetic life through things like hydrogen and one-carbon compounds like formate. And so when I talk about chemosynthetic life, I'm talking about organisms that use chemical energy as their primary source to produce biomass. And so as this kind of really brief equation is showing here, it's the hydration of ultramafic minerals and the production of hydrogen that's really important in this process. And so among the types of organisms that have really been the focus for trying to understand who these early potential analogs of life on Earth might be, acetogens and methanogens have been at the forefront of these discussions, and these are acetogenic bacteria and methanogenic archaea. And that's primarily because these organisms use these simple compounds, hydrogen and dissolved inorganic carbon, to produce biomass through simple enzymatic pathways, which I'll touch on a little bit throughout the talk. So what I would argue is that we're starting to learn quite a bit more about these types of organisms that are present in these environments, but we still know very little about their adaptations and how they're actually able to thrive in these particularly harsh systems. So the work I'm going to discuss today as part of my research extends out from what Alexis was talking about in the Somel ophiolite in Oman. And it's really an exemplary system for trying to understand microbial adaptations to serpentinite systems and the water rock interactions that occur there for a couple of reasons. One, it's really accessible. It's one of the largest near surface ophiolite systems on Earth. Two, there are wells that had been previously drilled there that tap into deep subsurface waters. And the third thing that I'm going to touch on kind of throughout the presentation is that there's a gradient in water types that are present in the subsurface of the Somel ophiolite. So there are waters that are essentially meteoric. These are slightly alkaline types of waters with pH maybe 8 to 9 or so. And as more extensive water rock interactions occur in the subsurface, these waters become progressively more alkaline, up to hyperalkaline types of subsurface fluids. And so this gradient really then allows us as microbiologists to understand the adaptations of organisms to this spectrum of conditions. And one thing to keep in mind that I'll touch on throughout the talk is that pH can serve as a sort of proxy for serpentinization reaction progress. So as more serpentinization influence, more water rock interaction occurs, waters become more progressively alkaline, as I mentioned. So my work that I'm going to focus on today is looking at these unique, intriguing kind of organisms that we found that were dominant in these subsurface fluids that belong to an uncultured candidate division called the acetothermia. Previous work by Katie Rempher in Alexis's lab showed that these organisms were particularly dominant in some of the most hyperalkaline fluids in the Somel ophiolite using 16S RNA gene-based studies. And so what I wanted to do, extending on from these initial finds, is use a genomic approach to try to understand what the adaptations of these organisms are using recovered genomes. And that's what I did. So I used a genome-resolved metagenomic approach to recover 7 different genomes from wells in the Somel ophiolite from different years and from different wells. So what's shown here on the right-hand side is just a phylogenetic analysis showing that there's 2 different types of these acetothermia that are present in these subsurface fluids. There's type 1, which I'll show in orange for the rest of the talk, and there's type 2, which I'll show in blue for the rest of the talk. The type 1 are more related to subsurface type organisms that have been found in hot springs, other subsurface systems around the globe, whereas the type 2 are really only related to one other population that was observed in the Lost City hydrothermal system, which, as Deborah Kelly talked about earlier, is a model, a serpentinite-hosted system. So two taxonomically very different types of organisms, probably different orders of acetothermia. In addition to being taxonomically very different, they also inhabit different ecological niches in the Somel ophiolite along water gradients. So the type 1, shown in orange again, pretty much only inhabit lower pH types of subsurface fluids, whereas the type 2 in blue again pretty much only inhabit higher pH hyperalkaline fluids in the Somel ophiolite. These fluids have been referred to previously as type 1, lower pH, and type 2 as higher pH, and that's actually kind of why I named these different populations as they are. So what that graph there is shown on the right is just an estimated relative abundance of these 2 different types of acetothermia in different subsurface well water communities. So each one of those different rows is a different community. The number in parentheses is the pH, and they're organized by the type of rock setting as well as their pH in ascending order. And so you can see that they pretty much have distinct distributions along the gradient of water types, which again, pH is a proxy for serpentinization influence. There's really only one community that had any bit of overlap between these two types, and it was a sample that was taken nearer to the surface from this NSHQ-14 well from a 50-meter sample. I won't talk about too much of the details of this, but there's evidence that there's mixing of near-surface waters in that particular sample that you don't find in the deeper sample that was taken from 85 meters, where you find essentially only the type 2 population. And so what these results suggest is that these type 2 acetothermia are much better adapted to these deeper hyperalkaline waters that are highly influenced by serpentinization. So what I really wanted to get at in my work is how these organisms are potentially adapted to these environments. And so to do this, I used a metabolic modeling based off of the genomes that I was able to recover. I don't want to scare you away with this graph that has a bunch of details on it. I'm just going to talk about some of the more prescient couple things here in turn. And so as I walk through these next couple of slides, you don't really need to worry about the enzymatic details of these various reactions. The type 1 enzymes are always going to be in orange, type 2 are always going to be in blue. One of the most important things that I was able to identify is that they both encode the capacity for autotrophic acetogenesis using the Wood-Lungdahl pathway. You might also know this as a reductive acetyl-CoA pathway. It's been invoked as one of the earliest microbial carbon fixation pathways in a lot of early life research. They both do this. Interestingly, they do it through different enzymatic complements, suggesting that they probably came about this capacity through different evolutionary trajectories. It's important to note that the Wood-Lungdahl pathway itself is your initial carbon fixation step, but it actually takes a bunch of more steps in order to get to larger biomolecules like sugars. Indeed, these organisms, both of them were able to encode the capacity for autotrophy via gluconeogenesis, suggesting that they were indeed capable of autotrophy. There's more differences in terms of what they're capable of doing. These type 2, again, these are the ones that you find in the most hyperalkaline fluids, appear to be primarily reliant on hydrogen gas for their energy conservation pathways. They encode electron bifurcating hydrogenases. These are used to produce reduced ferredoxin. Ferredoxin is like a low potential electron carrier. And that then is used to drive a chemiosmotic potential through an RNF complex. And that chemiosmotic potential can be used for ATP synthesis through an F-type ATP synthase. I should note that these complexes are essentially prevalently found in canonical model acetogens like Acetobacterium and the rest. So pretty consistent with what decades and decades of research of acetogens have uncovered as their energy conservation pathways. Type 1, on the other hand, do not apparently encode the capacity to use hydrogen gas. They might produce hydrogen through a biosynthetic pathway. They are able to produce a chemiosmotic potential, but through a totally different means. They don't encode RNF. They encode a bunch of membrane complexes that apparently allow them to use other oxidants for energy conservation, like nitrate, nitrite, and potentially oxygen. And they use a totally different type of ATP synthase to produce ATP. It's a V-type ATP synthase. And so taken together, these results suggest that these type 2 acetothermia are much better adapted to hyperalkaline waters that are highly influenced by serpentinization. For instance, they're pretty much reliant on hydrogen gas, which again is abundant in these types of environments. Whereas these type 1 acetothermia that we find in the lower pH, more oxidant replete waters appear to be capable of primarily using these oxidants that you find in more abundance there. So this is, one of the main goals of this work was to try to understand the characteristics of these organisms that allow them to inhabit these environments. But another aspect of it is to try to understand what they can tell us about the earliest life on Earth and the potential for life on other systems based on these adaptations in their evolutionary histories. And so I undertook some additional phylogenetic analysis to try to get at this question. First question I wanted to ask is where do they fall out on a tree of all of known bacterial lineages? So I undertook a whole bacterial domain genomic analysis, which is what's being shown in this large tree here. Don't really need to worry about any of the names. I'm just pointing out with that arrow where the type 1 and type 2 acetothermia fall out. Long story short, they're essentially some of the deepest branching groups that have been discovered yet, sister to others groups, the Thermatoga synergistedes and Deinococcus thermus, that a bunch of other studies have suggested are the earliest bacteria to have evolved. So very early branching. Is very highly supported branching placement in the tree as well. And the last little bit that I'll leave off of that provides some sort of intriguing questions regarding the potential for these organisms to serve as analogs for early Earth is the key acetogenesis genes that they use for the Wood-Lung-Dahl pathway. So these are carbon monoxide dehydrogenase acetyl-CoA synthase genes, or CODH ACS. These types have very different types of CODH ACS. The type 2 encode an archaeol-like system, whereas the type 1 encode a bacterial-like system. What I'm showing here is a phylogeny of just the archaeol-like. The bacterial one isn't quite as interesting, so I can talk about that later if you're interested in it. What's interesting about the type 2 CODH ACS is that it's pretty rare among bacteria, but I'm drawing your attention to where these type 2 complex subunits fall out in the tree with this red star. Essentially, these type 2 organisms, the CODH ACS they encode, are related to others that are found in serpentinite systems that have been found at the Cedars, for instance, as well as Lost City system. And they have a highly supported grouping with other type 1 methanogens, which have been thought to be some of the earliest types of methanogens, very well supported by the phylogenetic analysis. So these results potentially suggest that there was an ancient transfer of an archaeol-like CODH into subsurface bacteria, and particularly the types that are found in these serpentinite systems, which is an intriguing angle to follow up on. So to wrap this all up, these type 2 acetothermia in particular are highly abundant in the Semel-ophiolite subsurface waters that have the highest evidence for serpentinization influence. They may be key primary producers in some of these deepest waters and potentially in other serpentinization influence waters globally, like the Lost City hydrothermal system, for instance. I've shown a little bit of phylogenetic evidence that suggests they're among the earliest evolved bacterial acetogens known, and they exhibit evolutionary signals of deep ancestry of these important key metabolic genes like CODH ACS. And I would argue that these are some of the best so far characterized analogs of acetogens that have been hypothesized to be among the earliest to evolve on Earth in serpentinite environments. And they're a good system for future analysis. These are, of course, mostly based off of genomic data. We still have ongoing cultivation efforts in the lab to try to produce cultures of these type 2 acetothermia and the type 1 for that matter. Been able to generate some acetate-producing cultures from these hyperalkaline waters, but transfer is a problem with them. And so it's something we're currently working on in the lab. And so with that, I'd like to thank all the collaborators. This work was done in collaboration with Alexis's lab at UC Boulder, as well as John Spear at Colorado School of Mines, and all the folks that have helped out in the field and other aspects of the study. And I'd be happy to take any questions if there's time for that.
Thank you, Dan. We have time for one question.
Hi, Dan. This
Hi.
is Tristan Kara from CU Boulder.
Hey.
Great talk.
Thanks.
I'm curious if you have insights into what you think the primary carbon source that these organisms use is, because as Alexis talked about, these fluids can often be really DIC limited. And so do you think that's where the CODH comes in, or are there formate related proteins?
Yeah.
that you're trying to find?
Yeah, no, great question. As Alexis alluded to, DSC is a problem if you're a microorganism trying to be an autotroph in these systems. I think it's probably formate. They have, I didn't talk about it just for brevity, but these type 2 that live in these really hyperalkaline fluids have reversible electron bifurcating formate dehydrogenases, so they can interconvert between CO2 and formate pretty readily. I think that's probably the best guess for what they're using.
Thank you.
Yeah. Yep.
I have a question.
Oh, sure.
So Alexis touched on this a little bit, and I've heard about it from other people, that the contact between the periddotites and the gabbros are, sort of a hot spot, and certainly the cell members indicate that. I'm curious about the acetogens and if they're playing a role along that gradient. But what is it? I guess the question is, what is it about the gradient and the contact that makes it so special?
Yeah, that's a good question. And I think, just from my perspective, I think the evidence suggests that it's probably mixing of different water type fluids, right? From that talk about Prony Bay and some of the others as well, it's really the mixing of two different fluids where you get reductants and oxidants. And, once you have that combination of things, things can just totally take off in terms of what's capable in those systems. Personally, I think that's kind of what's driving a lot of the productivity in those sort of mixed contact types of settings. And I think Libby alluded to that in some of her work as well. And probably why you don't get as much productivity in the deeper waters where there's no mixing, right? It's just highly reducing. There's no oxidants. you know, you have things that are probably doing these really simple pathways where there's just no oxygen that's really available to drive anything. Yeah.
Thank you very much.
Yep.
All right, that concludes our session. Thank you all for attending, and thank you again to our speakers. The fun doesn't stop here, though. We have another online session at 2:30, so I encourage you to come to that. And then we also have a poster session. We have a lot of posters, and they're all awesome, and that's at 4:30 today. So thank you all for coming. I hope to see the rest of you at those sessions.